All right, kids, we're going to start today with kind of where we left off yesterday in lecture, and might go back just a little bit because I'm not for sure where both classes or where each class got to. But three major areas of the cell. And so if you can go faster through this, you can go ahead and advance it, or if you need a little bit extra time to finish writing, you can go ahead and do that also. But the three major areas. So basically, we have the outside and the inside and the in-between part. And the outside is the plasma membrane or cell membrane made of phospholipids, which we talked about last chapter. Then we have the in-between part, which is really where all the action happens. That's where proteins are made and things to be sent out of the cell are made. And then we have the brain of the cell, the control center of the cell, sometimes referred to, but at any rate, it's where the DNA, the control of the cell exists. So plants, in addition to the cell membrane, also then have a cell wall. Cytoplasm has all the organelles, and the organelles are the parts that we're going to spend some time talking about in a little more detail. And then the nucleus is in control of the characteristics and functioning of the cell. All right, structural features of the nucleus. What's going on in the nucleus? Well, the DNA, when the cell is not dividing, the DNA is in a configuration that it's in a configuration that we would call chromatin. And so that chromatin would be like this kind of a loose piece of string, right? And the reason that that's significant is all of these, every little segment of this string, in us, there might be as many as 20,000 different little segments of this string that each codes for something. You know, this might be what gives you type A antigens in your blood. And this might be what gives you a dark pigment in your hair. And this might be what codes for one of the enzymes. This might be what codes for one of the enzymes in your stomach that allows you to digest a particular nutrient molecule. So it's important that it's in this configuration when it's just being used. But when it's time for it to divide, it says chromatin coils into rod-like chromosomes. And so that looks more like this. Instead of that loose configuration, now we have something that looks more like this. And the reason that that's important is now it's easier to split that into kind of equal parts after it duplicates, to split it into equal parts so that each new cell has all of the appropriate information. All cells have the same type and the same amount of DNA. What we're saying by that is, you may have thought, well, in my skin, that's going to have DNA that tells my body and my cells how to make skin cells. And in my heart, that's going to have cells and information to tell me how to make cardiac muscle cells, which is what exists there. But the truth is that cell in your heart that's telling your heart how to make cardiac muscle cells also has the information for your skin and for those digestive enzymes and for the development of the nervous tissue in your brain and all of it. Every cell has every bit of information. And I think that's a real misconception that a lot of us have, and I think it's for good reason. It would seem that a particular cell would only have the information pertinent to that cell, but every cell has all the information. So then it becomes a thing which is a real point of research in today's medical world, and that is, how do those particular genes, like I was showing you on that string, how does that little piece of string get turned on? Like, how does your heart know to turn that part on that tells it how to make cardiac muscle cells? Or how does the cell in your skin know to turn on that little piece of thread that has the coding for how to make a skin cell? And how are all the other ones all turned off? Because if somehow that could be manipulated or controlled, things like spinal cord damage where your nervous tissue won't grow back, if someone could determine how to turn those genes on, maybe some of that nerve tissue, some of that sensation could be regenerated. All right. Then the other thing within the nucleus is nucleoplasm. And the analogy would be nucleoplasm is to the nucleus what cytoplasm is to the cell. So it's the fluidy part where, in the case of cytoplasm, it's where a lot of the metabolic activity takes place. Okay, so some other structures within the nucleus. The nucleolus or nucleolus, which is the dark area in the nucleus. So we have this dark area in the cell, that's the nucleus. And then we have this dark area within the dark area, that is the nucleolus, which is where ribosomes are made. Then the nuclear envelope is the part surrounding the nucleus. The nuclear envelope is to the nucleus as the plasma membrane is to the cell. And I was going to see if I could show you a picture of that. I think I can. Yeah. So let's take a brief look at that. And what we have there is on page 52 in your book. So this is showing that, and this is that nuclear envelope. And since I have it open, I'll go ahead and show you. There are these nuclear pores, which are these little dots here, or these openings, so that larger things could move in or out of the cell, particularly when we're making ribosomes, which are made in the nucleolus, but operate out in the cytoplasm. They have to be able to get out of these nuclear pores. So we have an inset right here. And then we have an electron micrograph that's showing with some clarity what's actually going on. And this is more of a real picture. This obviously is just a depiction or drawing or diagram. This is actually an image, an electron micrograph image. Okay. All right. So that nuclear envelope has pores, and as I said, proteins can move out of it and ribosomal subunits. I'm going to move through this pretty quickly. If you didn't have time to write it all, just pause the video and get caught up on the writing and then resume it. But the nuclear envelope, just like the plasma membrane, is a double membrane. That is, if we think about the phospholipid bilayer, it looks like this. There's a head on the outside of the cell, and there's a head on the inside of the cell, and then the tails face each other. And so it's a phospholipid bilayer. We also, I should have pointed that out better, but you'll see on page 50 and 51, or excuse me, 52, that is continuous. The endoplasmic reticulum is continuous with the nuclear membrane or nuclear envelope. Okay, ribosomes. Ribosomes get instructions from the nucleus, and they're actually what are making the proteins. And we hear all the time about DNA and DNA's critical role in how something is, how something's made, how something is. But it's important for us to understand, how is it making that happen? So if we said, well, somebody got the gene for type A blood, well, what does that mean? What does it mean to have type A blood? Well, it means that you have a protein kind of sticking out off of your red cell. Well, this is the red cell, and there's this particular protein sticking off on the outside that would be type A. And there might be a different shaped piece sticking off a different part of the red cell, and that might be a type B antigen. And anyway, but how does that happen? So let's say that, somebody says, well, I got type A blood from my mom because she had type A blood. Well, what it means is that strand of DNA or that gene that someone received from their mother coded for that protein that sticks off of the red cells. So the way it goes from being the code in the piece of string, like I showed you earlier, to actually what it is on the cell is by production of these proteins, protein synthesis. Ribosomes are made of two subunits. There's a big subunit, which is cleverly called the large ribosomal subunit, and then a smaller one that's, again, cleverly named the small ribosomal subunit. And those ribosomes are either scattered throughout, or they're sometimes found in a chain that looks like a bunch of pearls or beads. And they're also found on the membranes, on the endoplasmic reticulum, which means it's rough endoplasmic reticulum. So let's take another look at what that means. So those ribosomes, if I can find a good diagram of the ribosomes. So here, in this diagram, which is on page 50 of your book, you can see that we have all of these scattered ribosomes, all these little tiny dots, ribosome, 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 so on and so on and so on. And then we also have this rough endoplasmic reticulum, which is, as I told you earlier, continuous with the nuclear envelope. So stuff can move in or out of the nucleus and right onto this endoplasmic reticulum, which is kind of a passageway. But there's another interesting dynamic, and that is this polyribosome. So if you see right here, if your book opened up to that page, or down here, you have this thing that looks like a string of beads or pearls. And the reason that that's important is that allows for, well, let's go back to the original one. If we had a piece of DNA and it was coding for a particular protein, let's say that it was coding for a protein that we needed in our blood to create a clot. So that piece of RNA now, or that gene, would pass through this ribosome. It'd get clamped down and pass through this ribosome. And when it went all the way through, this ribosome would have produced a protein that was necessary for our blood to clot. Well, that's good and that's important. But think about when you have an injury and you need for that blood to be clotted. That'd be pretty painstaking if every ribosome just had to make one of those proteins when you need them quite rapidly. Well, see if I can get that, or I can get the polyribosome on here. Okay, so now let's say that instead of doing that, now we're going to use this polyribosome. We're going to use it instead of just one of these single ribosomes. Now, here's the analogy I'm going to make, and this may or may not make sense to you. Hopefully, I'll have an opportunity to explain it to you in person. But let's say that all of you, for homecoming, you were all supposed to bring a dozen cookies, and we wanted all the cookies to be the same. But we only had one copy of the recipe. Now think about this. Let's say that we had this one copy. And so we take this copy, and this is one of you. And you make a whole batch of cookies. So when you're finished, and not until you're finished, you hand that recipe on to this person. This is another one of you. Then they make their dozen cookies. Then when they're all finished, they hand it on to the next person, and they make a dozen cookies. Now, could we make that work? Did everybody make a dozen cookies using the same recipe? Well, yes, but what's the obvious problem? Well, it's going to take a really long time. It's going to take a really long time. Now, let's say, on the other hand, we had that one recipe, and instead of it being written like on a sheet, it's written on, you guys aren't going to know what I mean, but a ticker tape, where it's one thin, continuous instruction. You know, preheat the oven, obtain these ingredients, you know, put together the dry ingredients, add milk, blend, whatever, or mix, or whatever. Okay? Now, if we had it written out like that, one step after the other on a long strip of paper, what if we still only have one recipe? We still only have one recipe. But let's say that all of you are working side by side. And so now the first one of you starts. And so this person is preheating the oven. And then while they're gathering the dry ingredients, this next person reads the preheat the oven. So this one's preheating the oven. This one, and you're all working separately, but you're using the same set of instructions. This one's preheating the oven. This one's gathering the dry ingredients. Okay, now it moves on. Now this one's preheating the oven. This one's gathering the dry ingredients. This person is cracking eggs. And this next person is adding milk. Whatever it is, all the steps. So now when we get all the way through, okay, this person will be finished making their cookies. This person will be starting, and everybody in between will be somewhere along the line. But by the time we get to here, everybody will be done. Everybody will be done because we're using that same set of instructions. So here's the crux of the analogy, is that with one set of instructions, we're able to make all of these cookies much more quickly, because we really only need to see one part of the instructions at a time. And somebody else just as well be looking at the other parts of the instruction and continuing to do what they're doing. Now, let's go back to the actual application of this in cells. We're bleeding, we're hemorrhaging. We need to make a lot of clotting proteins in a real hurry. If we did it this way, there'd be a lot of blood loss before we had enough clotting proteins to get it to stop. But if we did it this way, now we're going to be able to make a lot more clotting proteins a lot faster. That's a polyribosome. It's used when you need to make a lot of something in a really short time. Okay, so now we're going to move to some of the other cytoplasmic organelles. And we're going to categorize them in three ways. Some of them are for getting and using energy. Some of them are for kind of moving things around and packaging things and so on. And then some other ones are for structure, just giving the cell its shape and allowing the cell to move, or even allowing things to move within the cell. Not the actual packaging part, that's here, but then this kind of lays down tracks for those things to move. Again, I'm going to remind you, if you need to pause this, just pause it, and then you can move on when you're ready. Energy organelles. And we only have one of these, chloroplasts or mitochondria. We only have one of them. We only have mitochondria. And because we get our food, if you remember from a couple chapters ago, we get our food by ingestion. We're not making our own food. We can kind of remanufacture it. We can take what we eat and break it down and build it back up, but we can't start from scratch and make our own food the way that plants can. So plants have chloroplasts for making their own food. They actually also have mitochondria, because not only do they need to make food, they also need to get energy out. But chloroplasts undergo photosynthesis, and that solar energy is stored in carbohydrates. You see if you remember this. Somebody go ahead and say it out loud. What's the stored form of carbohydrates in plants? The answer to that is starch. Starch. Okay? then mitochondria take starch or other such nutrient molecules and break it down via a process called cellular respiration. And in that process, the chemical energy, which is the energy in the bonds that are holding the starch or sugar together, those are broken. And so you may or may not be aware, but in order for bonds to form, it requires energy. But then when those same bonds are broken, energy is given off or released. So that chemical energy that's captured from the breakdown of carbohydrates is stored short term in a molecule called ATP, adenosine triphosphate. And that adenosine triphosphate means it has 3 bonded phosphate molecules. But when it's going to give off its energy, it loses one of those and energy is given off. And then when more energy is available, it's built back into adenosine triphosphate. So this molecule just keeps going back and forth. Energy's added, it becomes this. If energy's needed, it gives off energy. Energy's added back to it, goes back to this. Energy's given off, goes back to that. Mitochondria. You may have heard somebody say in sincerity or maybe even in jest, the mitochondria is the powerhouse of the cell. But it truly is the powerhouse of the cell. The mitochondria is the part of the cell that releases energy from foodstuffs. And so the energy, it'd be the equivalent of the engine in your car. You have fuel, and the energy's in the bonds of the fuel, but you have to have a way to get it out. And the way you get it out and convert it to a usable form is in the engine of your car, right? The fuel gets burned, and that energy gets captured and converted into movement of the car down the road. So the mitochondria is kind of like the engine of your car. It's converting those bonds that are in gasoline, or in the case of your body, those bonds that are in glucose. It's breaking those open and then availing the energy that your body needs, or in the case of your car, that your car needs to move. It's making that energy available to do just that. So they're double membrane. So remember, the cell is double membrane. It's like this, phospholipid bilayer. But the mitochondria is also double layered. And I'll show you that just briefly here. That's this diagram. So you might want to flip to page 57, it looks like. And here's that diagram on page 57. That where you can see it. So here are the parts of the mitochondria. So it has, this is one of the membranes, and then the inside membrane has these curves and twists in it. And that's called the cristae. The inner membrane is called the cristae. And then the fluid inside is called the matrix. So we have an outer membrane, an inner membrane, and then the fluid inside, which is called the matrix. So that fluid matrix contains the DNA, what's called, you may have heard this in the news before because they've figured out ways to use it. It's called mitochondrial DNA, and then ribosomes and enzymes. And in fact, we'll get to toward the end of this chapter the fact that there are biologists and scientists who believe that mitochondria may have at one time been their own independent bacterial molecule, and only more recently maybe got taken into another larger cell and became just a portion of the bigger organism. So there's no real proof of that, but they are very much like, they're structured very much like a simple organism, like a bacteria. Membrane folds on the inside to make cristae, and that's where hold the proteins that make ATP. And we'll spend a whole chapter on respiration. So we're not going to talk a lot about it now, other than to just say that's where it happens. That's where the real action is. Chloroplasts, of course, chloroplasts are only found in plant cells, algal cells, plants and algae. Simpler organisms like bacteria can even have thylakoids, which and enable them to use the sun's energy. They have chlorophyll, and chlorophyll is the pigment. Chlorophyll is the pigment inside of a plant cell. It's what makes it green. And it absorbs, that's the actual, chlorophyll molecules are actually what absorb the sun's energy. We say, well, plants need light to survive, or plants need light to, produce starches and so on. Very true. But the actual molecules that are collecting that energy from the sun are chlorophyll molecules. In fact, clusters of chlorophyll molecules called photosystems. Stacks of thylakoids in the stroma. Stroma is the fluid inside the chloroplast. They form grana. Stacks of them are called grana. And chlorophyll in the grana captures solar energy. Stroma has DNA, ribosomes, and the enzymes needed to make carbohydrates. Particularly for our purposes, we would say to make starches, make those big polysaccharides. So let's take a look at that. So here is a chloroplast. And here are some of the parts that are important. So we have these stacks. Well, first of all, let's say these discs. These individual discs are called thylakoids. These are called thylakoids. Then a stack of them looks like a stack of poker chips. That's called a granum. And then just like the mitochondria, there's a double membrane, an outer membrane and an inner membrane. And then inside are housed these thylakoids, which are in stacks called granum. And then the other critical term is stroma. So I want you to see if you can finish this analogy. The analogy is stroma is to a chloroplast as blank is to a mitochondria. Stroma is to a chloroplast. It's the fluid in the chloroplast. Stroma is to a chloroplast as blank is to a mitochondria. The answer to that is matrix. The fluid in a chloroplast is stroma. The fluid in a mitochondria is called the matrix. Okay. Guys, we're going to just get a little bit further into this, and then we'll call it a day. But now we're ready for the endomembrane. This isn't another organelle. This is another category of organelles. So the endomembrane system, and what that means is within the membrane, stuff that's happening within the membrane. Nuclear envelope, that's a barrier between the nucleus and the cytoplasm. The endoplasmic reticulum, or kind of those tracks, Golgi apparatus, and vesicles. Those are all parts of the endomembrane system. And that endomembrane system compartmentalizes the cell. It means it divides it into different sections. So the nuclear envelope divides the nucleus from the cytoplasm. The endoplasmic reticulum are the kind of these tracks that extend. Vesicles can fuse with the plasma membrane and release stuff to the outside and so on. Okay, so the first one, endoplasmic reticulum. And endoplasmic reticulum are channels that extend from the nuclear envelope. Rough has ribosomes. So rough endoplasmic reticulum means it has those little dots. It has those little ribosomes that function in making proteins. Proteins are made there and enter the endoplasmic reticulum and get modified. So there's kind of the initial manufacture of proteins, and then those proteins maybe need to be packaged or shaped differently before they're sent out. Then we have smooth endoplasmic reticulum, which is continuous with the rough endoplasmic reticulum. And it, instead of making proteins, smooth endoplasmic reticulum makes lipids or phospholipids. So see if I can find you a good diagram of that. Look on page, probably just the big diagram is probably as good as we can do on page 50. Okay, so look on page 50. I'll put it up here also. Okay, so this with the little dots on it is rough endoplasmic reticulum, and then it extends out here to this is smooth endoplasmic reticulum. So in here is where proteins would be produced. Out here is where fats or phospholipids would be produced. And then these little circles, if that stuff's being packaged to be sent out of the cell, those would be considered vesicles, well, packages. Okay. The next part is the Golgi apparatus, and I'm going to go ahead and show you that while I have this up here. This is the Golgi apparatus. Okay, it's the Golgi apparatus. So these proteins and fats and so on that were made can be packaged for being sent out. And again, these are all these vesicles. Those are things that are ready to be sent out of the cell, or packaged. So that's what the Golgi apparatus does. It takes in things, so it's kind of faces like this, so that things that are made by the ribosomes and by the smooth endoplasmic reticulum, fats and proteins can be packaged and processed for delivery outside the cell. So discovered by a guy named Golgi, made of a series of curved sacs. The interface, as I was just showing you, is directed toward the endoplasmic reticulum. The outside faces the outside of the cell because it's sending things out to be used in a different part of the cell. And then it processes and packages proteins and lipids for secretion, or just the purpose of being sent outside the cell. Okay, we have just three more screens here. We're going to finish it out. So push through here. Processes and packages to be sent out. So proteins that are needed outside the cell. Maybe it's lipids for the purpose of being part of a hormone or something like that, or proteins that are necessary to rebuild muscle. Okay, or proteins that are necessary, like we talked about before, for clotting or something like that. We have a structure called a lysosome, and they have hydrolytic enzymes. And hydrolytic enzymes break stuff down. So lysosome's job is to break stuff down. Digest waste, digest old, worn out cell parts, and autodigestion means self-digestion. So it's digesting stuff that's part of the cell itself, but maybe it's parts that are no longer needed or useful. And the inability of that to function properly can result in a lysosomal disease. And I'll read you a little bit about Tay-Sachs because it always comes up, and then people want to know a little bit more about it. I'm not going to give you the whole thing, but hopefully it'll give you a little idea of kind of what it's about. So Tay-Sachs is a genetic disorder that results in the destruction of nerve cells in the brain and spinal cord. Most common form is infantile Tay-Sachs. Becomes apparent around the age of 3 to 6 months, with the baby losing the ability to turn over, sit, or crawl. Then there can be, sensory problems, seizures, hearing loss, inability to move, and oftentimes causes a very early death, even age 3 to 5. Can occur later in childhood or even into adulthood, but tends to be less severe. Let's see if I can give you, caused by a genetic mutation in a gene that's on your 15th chromosome. It disrupts the activity of the enzyme build up and causing toxicity. So it just causes things to build up, which is logical enough considering that the function of this is to break stuff down. And so if those substances build up, it is problematic. Okay, vacuoles. Vacuole is larger than a vesicle. It's kind of a storage area. In plants, we can have something called a central vacuole. I'll show you that very briefly here. This is the central vacuole of a plant. So this central vacuole of a plant can actually fill up with water, and that's what makes a plant that's well watered, full of water, kind of rigid in its stem. And if it starts to lose water, it's what causes a plant to wilt. But animal cells don't have this giant central vacuole. Animal cells have vacuoles, but not a giant central vacuole. Also present in protozoans, contractile vacuoles in freshwater protozoans. Okay. And that contractile vacuole is an organelle in water regulation, and so allows water in and out of simple organisms, freshwater protozoans. All right, the last screen we're going to look at today has to do with peroxisomes. So a little bit like lysosomes, but differentiated by the fact that they produce a byproduct of hydrogen peroxide. And then that's broken down by the enzyme catalase. And Lorenzo's oil, there's actually a movie, I think, by that name about a child who had that condition. And I suppose the reason they call it adrenoleukodystrophy, ALD. Okay, but anyway, that's peroxisome disease. So you don't need to know a lot about it other than when these things don't, like anything in your body, when something's not functioning right, it has farther reaching effects than it just not working right. Everything needs to be in an extremely kind of delicate balance. All those things need to work together. All right. So for tomorrow, the questions that you should have answered, that you need to have answered, are 1 to 41 on pages 1 and 2. So really all of page 1 and 2. You need to answer questions numbers 1 to 42 for Thursday.